Reports from the pharmaceutical sector indicate a significant challenge in the global response to hantavirus, as existing vaccine candidates and current medical interventions appear unable to provide protection against a newly identified mutated strain of the pathogen. This development has triggered a reassessment of public health preparedness regarding zoonotic diseases, particularly those involving high-consequence viral pathogens. Hantaviruses are a family of viruses spread mainly by rodents and can cause varied disease syndromes in people worldwide. In the Americas, they are primarily known for causing hantavirus pulmonary syndrome, or HPS, a severe and sometimes fatal respiratory disease. In Europe and Asia, they are the primary cause of hemorrhagic fever with renal syndrome, or HFRS. The current reporting suggests that the genetic shift observed in this specific mutation has altered the viral structure to a degree that renders the current preventive measures, which were developed based on older or more common strains, largely ineffective. Understanding this threat requires looking at the fundamental nature of hantavirus transmission. Unlike many other respiratory viruses, hantaviruses are not typically spread between humans, with the notable exception of the Andes virus in South America. Instead, humans become infected through contact with hantavirus-infected rodents or their urine and droppings. The primary route of infection is the inhalation of aerosolized viral particles. When rodent waste is disturbed, the virus can become airborne and enter the human respiratory system. This mechanism makes the virus particularly dangerous in rural, agricultural, or abandoned industrial settings where rodent populations may go unchecked. The news of a mutated strain suggests a level of viral evolution that could potentially alter how the virus interacts with human cellular receptors or how it evades the initial immune response. Public health officials emphasize that while hantavirus infections are relatively rare compared to seasonal influenza, their high mortality rate, often exceeding 30% for pulmonary cases, makes any lack of vaccine protection a critical concern for global health security. As the scientific community analyzes the implications of this mutation, the focus remains on the geographic distribution of rodent reservoirs and the environmental factors that lead to human exposure. The framing of this issue by industry insiders highlights a gap in our current medical arsenal, pointing to the inherent difficulties in developing broad-spectrum protection against a virus family that exhibits significant genetic diversity. This situation serves as a stark reminder of the continuous evolution of zoonotic threats and the necessity for robust, ongoing surveillance to track how these viruses change as they circulate in wild populations. The reported admissions from pharmaceutical developers regarding the limitations of existing vaccine technology focus on the complex molecular architecture of the hantavirus. Genomic sequencing and laboratory analysis are the primary tools used by health agencies to monitor these developments. According to available data, the hantavirus genome consists of 3 segments of negative sense, single-stranded RNA, which are prone to mutations during replication. The specific mutation in question likely affects the viral envelope glycoproteins, known as GN and GC. These proteins are the primary targets for neutralizing antibodies produced by the human immune system or induced by vaccination. If these proteins undergo a significant structural change, the antibodies generated by older vaccine formulas may no longer be able to recognize or bind to the virus, allowing it to infect cells unimpeded. Medical clinics and specialized laboratories that handle high-consequence pathogens are currently the front lines for observing the clinical impact of these shifts. While some regions, particularly in East Asia, have utilized inactivated hantavirus vaccines for decades with varying degrees of success, these are often strain-specific. The current reporting underscores that a mutation in the wild could effectively bypass the protection offered by these regional products. Furthermore, in the United States and Europe, there is currently no FDA-approved vaccine for general public use, leaving the population dependent on experimental treatments and supportive care. Hospitals and intensive care units managing suspected cases rely heavily on molecular diagnostics, such as polymerase chain reaction or PCR testing, to confirm the presence of the virus. However, if a mutation is severe enough, it can occasionally interfere with the primer binding sites used in standard diagnostic assays, potentially leading to false negatives and complicating the clinical picture. Surveillance programs managed by organisations like the Centers for Disease Control and Prevention and the World Health Organization are tasked with tracking these viral variations in rodent populations before they spill over into humans. The logistics of this surveillance involve complex fieldwork, where biologists and epidemiologists trap rodents and test tissue samples to map the prevalence of specific strains. The admission that existing pharmaceutical research has not kept pace with these mutations suggests a need for a more adaptable platform for vaccine development, such as mRNA technology, which can be updated more rapidly than traditional protein-based or inactivated virus methods. As of now, the data indicates that while the mutated strain has been identified, the exact scale of its clinical impact remains under investigation. And health agencies are working to determine if the mutation also affects the severity of the disease or its mode of transmission. In light of the reports that current vaccine candidates may not offer protection against the mutated hantavirus, public health guidance continues to prioritise environmental management and exposure prevention as the most effective tools available. Prevention strategies are centred on the principle of minimising human contact with rodent excreta. Health officials advise the public to avoid stirring up dust in areas where rodents may have nested, such as cabins, sheds, or storage units that have been closed for extended periods. Instead of sweeping or vacuuming, which can aerosolise viral particles, the recommended procedure involves wetting the area with a disinfectant or a mixture of bleach and water to deactivate the virus before cleaning. The use of personal protective equipment, including gloves and high-efficiency particulate air or HEPA filtered masks, is strongly encouraged for individuals working in high-risk environments. Surveillance remains a critical pillar of the global response, with authorities monitoring both rural and suburban areas for shifts in rodent populations that could signal an increased risk of spillover. The honest limit of current knowledge lies in the unpredictability of viral evolution. While scientists can identify mutations in a lab, predicting exactly when or where a mutated strain will cause a localized outbreak remains a significant challenge. There is also a degree of uncertainty regarding how climate patterns, such as milder winters or increased rainfall, may influence the population spikes of carrier species like the deer mouse or the bank vole. For travellers and those living in endemic areas, the primary message from health departments is one of vigilance rather than alarm. Recognising early symptoms, which typically include fever, muscle aches, and fatigue, followed by a rapid onset of shortness of breath, is vital for improving clinical outcomes through early supportive care. While the pharmaceutical industry works to address the gaps in vaccine efficacy, the medical community remains focused on improving the speed of diagnosis and the quality of respiratory support in clinical settings. The current situation highlights a broader reality in virology. The viruses we track are constantly moving targets, and our public health infrastructure must remain flexible to adapt to new genetic data as it emerges. This report underscores the importance of continued investment in basic virology and environmental health to protect against pathogens that do not yet have a pharmaceutical solution. For more updates on emerging public health developments and surveillance reporting, consider subscribing to this channel. The current reporting from the pharmaceutical sector highlights a critical vulnerability in global immunologic defenses, specifically regarding the molecular evolution of the hantavirus genome. At the centre of this development is the realisation that the viral proteins targeted by previous vaccine research have undergone significant structural changes. Hantaviruses are enveloped, single-stranded RNA viruses with a genome divided into three segments: small, medium, and large. The medium segment, or M segment, is of particular concern to researchers because it encodes the enveloped glycoproteins known as GN and GC. These glycoproteins form the external spikes of the virus, which serve as the primary mechanism for attaching to and entering human cells. Most experimental vaccines and existing regional treatments are designed to prompt the human immune system to produce neutralizing antibodies that recognize these specific spikes. However, the reported mutation suggests a level of antigenic drift, a process where the virus accumulates genetic changes that effectively reshapes these glycoproteins. When this occurs, the antibodies generated by older vaccine formulas find no recognizable binding site on the mutated virus, rendering the immune response ineffective. This phenomenon, often referred to as immune escape, is a primary reason why pharmaceutical developers are admitting that current stockpiles and late-stage candidates may not provide the anticipated level of protection against this emerging variant. The admission underscores the technical difficulty of creating a universal hantavirus vaccine. As the family of viruses exhibits high genetic diversity across different geographic regions and rodent reservoirs, the institutional response to this mutation involves a complex coordination between private labs and national health agencies. Because hantaviruses are classified as high-consequence pathogens, all laboratory work involving the live virus must be conducted in Biosafety Level 4, or BSL-4, facilities. These are the most secure laboratories in the world, equipped with specialized ventilation and rigorous decontamination protocols. In these settings, virologists are tasked with conducting cross-neutralization assays. These tests involve taking blood samples from individuals who have received experimental vaccines or who have recovered from older strains of the virus and exposing those samples to the newly mutated strain. If the antibodies in the blood fail to stop the virus from replicating in a lab dish, it confirms that the mutation has successfully bypassed existing immunity. The data currently emerging from the sector suggests that this is exactly what is happening. Furthermore, the pharmaceutical industry faces a significant logistical and financial hurdle in retooling production lines. Unlike modern mRNA platforms, which can be reprogrammed relatively quickly by changing the genetic sequence used in the vaccine, older inactivated virus technologies require growing large quantities of the live virus in controlled environments, a process that can take months or years to refine for a new strain. This delay between the identification of a mutation and the deployment of a corresponding medical countermeasure represents a significant gap in global biosecurity, beyond the challenges of vaccine efficacy. The mutation poses significant hurdles for diagnostic surveillance and hospital labs. Most rapid diagnostic tests for hantavirus rely on identifying specific antibodies or using polymerase chain reaction, known as PCR, to find viral RNA in a patient's blood. If a mutation is extensive enough, it can lead to what scientists call diagnostic escape. This occurs when the molecular primers used in PCR tests no longer perfectly match the viral sequence, leading to a failure in amplification and resulting in false negative results. For clinicians in high-risk areas, this means that a patient could present with all the hallmark symptoms of hantavirus pulmonary syndrome, such as fluid in the lungs and sudden respiratory distress, while the laboratory results remain inconclusive. This uncertainty complicates the clinical management of the disease, as there are no specific antiviral drugs currently approved for hantavirus. Treatment is primarily supportive, involving mechanical ventilation and careful fluid management. The reported failure of existing vaccines to stop the mutated strain places an even greater emphasis on these diagnostic tools, as they are the only way to confirm the movement of the virus through a population. Consequently, health officials are calling for an immediate update to genomic sequencing databases to ensure that labs worldwide can identify the unique signature of the mutated strain. The situation remains fluid as researchers wait for more clinical data to determine if the structural changes in the virus also correlate with changes in how the disease progresses once a person is infected. With the pharmaceutical sector confirming that existing vaccine candidates are currently ineffective against the mutated hantavirus strain, the global public health strategy has pivoted toward intensified surveillance and rigorous environmental prevention. Since no medical shield is currently available, the primary defense remains the interruption of the transmission cycle between rodents and humans. Health agencies emphasize that the most critical prevention measure is the proper management of environments where rodent reservoirs, such as deer mice, white-footed mice, or rice rats, are known to thrive. Public guidance remains steadfast on the dangers of aerosolization. When rodent urine or droppings are disturbed, the virus can become airborne in microscopic droplets. Inhaling these particles is the most common route of infection. Therefore, officials strictly warn against dry sweeping or vacuuming in areas with evidence of rodent activity, such as seasonal cabins, tool sheds, or crawl spaces. Instead, the recommended protocol is a wet cleaning method, saturating the area with a disinfectant, specifically a mixture of 1 part bleach to 9 parts water, for at least 5 minutes before wiping. This ensures the virus is deactivated and remains trapped in liquid rather than entering the air. For individuals in high-risk occupations or those performing deep cleans of infested areas, the use of professional-grade respirators such as N95 or P100 masks is considered a standard safety requirement. The limitations of current knowledge regarding this mutation necessitate a robust surveillance framework known as the One Health approach, which links human, animal, and environmental health data. Epidemiologists are closely monitoring the population dynamics of carrier species, as environmental factors like heavy rainfall followed by drought can lead to an explosion in rodent populations and a subsequent increase in human exposure. The current uncertainty lies in whether this specific genetic mutation alters the virus's environmental stability, essentially, how long it can survive outside a host. While most hantaviruses lose infectivity within hours or a few days when exposed to sunlight and dry conditions, any change in this duration could significantly impact the risk profile of contaminated buildings. Furthermore, the global health community is focused on the ethical imperative of transparent data sharing. Because hantavirus disproportionately affects rural and agricultural workers, as well as marginalized communities with poor housing infrastructure. There is an ethical demand for equitable access to diagnostic tools and future therapeutic developments. Reporting indicates that the pharmaceutical industry must balance proprietary research with the public need for rapid genomic updates to diagnostic kits, ensuring that no region is left behind due to a lack of updated testing primers. Practical takeaways for the general public include a focus on exclusion and early symptom recognition. Excluding rodents from living spaces involves sealing any holes or gaps larger than 1/4 of an inch, roughly the size of a pencil, using steel wool or metal flashing, as rodents cannot easily gnaw through these materials. Additionally, maintaining clean outdoor perimeters by removing wood piles and tall grass near home foundations can reduce the likelihood of nesting. From a clinical perspective, since there is no vaccine-induced protection, the importance of early intervention cannot be overstated. The initial symptoms of hantavirus infection, which include fever, severe muscle aches in the large muscle groups, and fatigue, can easily be mistaken for more common viral illnesses. However, if these symptoms are followed by a sudden onset of shortness of breath or a dry cough, immediate medical attention is required. Physicians must be informed of any potential rodent exposure to facilitate rapid testing and supportive care, such as oxygen therapy or extracorporeal membrane oxygenation in severe cases. As the scientific community works to bridge the gap between viral evolution and medical countermeasures, the current situation serves as a reminder of the persistent challenges posed by zoonotic pathogens. This report